Greetings to all. Myself, Dr.P.Samuel, Assistant Professor of Biotechnology, Bishop Weber College, Trichy. Today the topic what we are going to discuss is quorum sensing in bacteria. The reason behind choosing the topic is, do bacteria communicate with each other? It is very essential to understand, like human beings, bacteria also communicate with the neighboring or other group of bacteria, and this phenomenon is said to be quorum sensing, and the mechanism and what is the outcome of this mechanism will discuss in detail. We'll move to the first slide. What is actually quorum sensing? By definition, it is a mechanism adopted by bacteria to communicate and coordinate behavior based on cell population density. So it is based on the population of cells in a micro environment. Micro environment may be inside the body, or for example in our mouth. Mouth itself called as a very good environment for the growth of bacteria. And here it is actually based on the population density. This is what the keyword given per the definition. It uses signaling molecules. It uses signaling molecules called autoinducers, and these signaling inducers or these signaling molecules are actually produced by bacteria, which will regulate behaviors like biofilm formation, virulence and bioluminescence in the neighboring population. So a response is given by one bacterial cell. The response is accepted or recognized by the neighboring cells. In response to the signal, the neighboring cell will react. The reaction given by the neighboring population is called as either biofilm formation or virulence or bioluminescence. And this is a small illustration with respect to quorum sensing. Imagine this is one group of bacteria, right? The one which you find on the left hand side, one group of bacteria. This group of bacteria will produce signals, and these signals are called as autoinducers. It differs according to the type of bacterial population. Not all bacteria will produce same type of autoinducer or signals, and these autoinducers, right, will passed on to the neighboring cells, right? This is another group of bacteria, right? And this bacteria will accept the signal produced by one population of bacterial cells. In response to the acceptance of signal, it started to respond accordingly. And for example, this is an illustration for formation of biofilm. The bacterial population which accept the signal will start to form biofilm. Biofilm refers to encapsulation of bacteria in a matrix. In a matrix synthesized by the own bacterial population, right? So it produces one group of bacterial population produce signal, first thing, and the signal produced by another, the neighboring bacteria will accept the signal, and in response to acceptance, it gives or it forms biofilm. This is what the coordination between the bacteria in response to quorum sensing. Right, we'll move on to the next slide. How quorum sensing works, how it works. So to begin with, as we discussed in the previous slide, one group of bacteria produce a chemical called autoinducers, right? Here these autoinducers accumulate in the environment. For example, if we take mouth is the microenvironment for bacteria, this autoinducers will accumulate in the mouth, and what happens as a result? what would be the result of accumulation of these signals? This has to be discussed. After accumulation, it has a limit called threshold concentration. Threshold concentration refers to a limit. When the limit is reached, when the concentration of autoinducer in the microenvironment is reached, it binds to the receptor. This is to be noted. Receptor, there are proteins expressed by the neighboring bacterial population. And upon binding of the autoinducer, it activates, the receptor activates or triggers the gene expression, which will end up in the response given by the neighboring bacterial population. So this is very simple mechanism, but it has to be studied and it has to be understood by each and every one of us to understand how bacteria communicate with each other. Next slide. And this is the illustration of the previous slide. This bacterial population produce a lot of signals called autoinducers. These autoinducers will accumulate in the environment, for example, our mouth. Once the concentration is reached to a maximum limit called the threshold limit, it will bind to the neighboring bacterial cell, and according to the binding, the receptor gets activated. Receptor activation will result in the response given by the neighboring bacterial cell. This is also a small illustration, and these molecules are called as signals, and this is the neighboring bacteria, the one which is accepting the signal. And these are all the common chemical signals produced by different groups of bacteria. In case of gram negative, it produces acyl homoserine lactones. In short, it is called as AHL. And gram positive bacteria use oligopeptides. So I am giving small sort of representation, diagrammatic representation, so that you people can understand very easily. And the universal signal, irrespective of gram positive and gram negative bacteria, the uniform or the universal signal produced by almost all group of bacteria is AI2. AI refers to not artificial intelligence. AI refers to autoinducers 2. And this is for interspecies communication. This is species number 1 and this is species number 2. Means this is one group of bacteria and this is another group of bacteria, but irrespective of their origin, irrespective of their domain, irrespective of their genus, it produce or it recognize or it exchange a specific molecule called AI2, which will help the other bacteria to respond. Next slide. And these are all some notable systems, right? In organism like Vibrio fischeri, this is a well-known fish pathogen, and this will produce, respond to the autoinducer called bioluminescence, and this can be well studied in marine aquatic ecosystem. Bioluminescence can be observed in Vibrio fischeri. Pseudomonas aeruginosa is a human pathogen and well studied pathogen also. It regulates biofilm formation and virulence factors. And third thing is Staphylococcus aureus. This is also a very common human pathogen, and it regulates the toxin production, the response, by a system called AGR system. AGR system. We'll move on to the next slide. And quorum sensing and biofilms. See, what is biofilm? It is a structured bacterial communities encased in a self-produced matrix. And this is like we are encasing egg into a box, right? The box contains different numbers of eggs in a safe and protected form. Like this, in biofilms, bacteria will be encased within self-produced matrix. And quorum sensing will promote or it will regulates adhesion, matrix production and detachment for dispersion. So these are all the different roles of quorum sensing in controlling the communication between different bacterial forms. And why do bacteria use quorum sensing? Another thing. So we studied mechanism and we studied how it is regulated, and why do bacteria use quorum sensing is another important thing. The thing is cooperation, it enables collective behavior. To survive, all the bacteria will function in a coordinate form, right, cooperative form. So for that it has to produce or it will use quorum sensing. And resource optimization, it avoids wasting energy. Bacteria, they are so stringent, they do not waste their energy like we waste unnecessarily, right. They are very stringent in utilizing energy. So quorum sensing will also allow the bacteria to utilize or to spend the energy in an efficient way. And in defense mechanism, this is another thing, right, how it escapes from the weapons used by human beings like antibiotics, right. So bacteria will produce these matrix which will protect the organism from substances like antibiotics and host response like immunity. And why quorum sensing matters? Medical application, industrial application and environmental application. Why we need to discuss or why we need to understand these things? Medical application, we can produce novel antibiotics targeting the quorum sensing proteins, another one important thing. And for industrial application, we can promote what substances can be identified in the laboratory can be implemented in the industry, so that it will promote the formation of biofilm which will help in industrial process like wastewater treatment. And in environmental application, it is also adopted by the bacteria to promote bioremediation strategies. And quorum quenching, disrupting communication. Once we disturb the process or the mechanism, the communication between bacteria will be definitely disrupted. For example, interfering with quorum sensing signals to inhibit bacterial communication. Bacterial communication. If we want to communicate with another person, for example, if another person whom I want to talk to is wearing an AirPod, in the sense I couldn't able to communicate with him. In the same way, we can inhibit or we can interfere into the communication between bacteria through a process called quorum quenching, right. So methods like enzymes degrading autoinducers, signal analogs blocking receptor binding, natural compounds from plants or fungi. So these are all the agents which will help us to interfere with the communication between the bacteria. Once the communication is disturbed, one bacteria do not know how to cooperate or do not know how to interact with another bacterium. If the interaction is prevented, it cannot able to exhibit the secondary or tertiary response, right. Hope you have reached the last one. Summary and outlook. So summary in the sense, the things whatever we discussed in the class, just recall all those things. What is quorum sensing? What is the mechanism of quorum sensing? How quorum sensing works? And what is called quorum quenching, everything. Summarize all these things. And quorum sensing is central to bacterial cooperation and survival. And research. As you people are an active and enthusiastic students, basically interested in research, you should understand, you should take this area as a interest, so that you can understand mechanism in diverse bacteria and developing quorum sensing inhibitors as therapeutics. This is one such field to explore a lot, right. So, or in other words, this is one such field yet to be explored a lot, right. So the final thought is, exploring quorum sensing provides insights into bacterial life and strategies to combat infection. Thanks for the opportunity given. For any queries, please.